So, good evening, participants. I welcome you all in this Science Dialogue series. And as you know, today's speaker is Dr. Atul Tiwari ji. He has joined with Sivani Pandey,
Not
name
Shivani sir, Shivam Pandey.
Shivam Pandey. Okay, Shivam Pandey ji. But his name is Dr. Atul Kumar Tiwari. So, I welcome you, Atul Kumar ji,
Thank you,
in
sir.
this Science Dialogue series. And thank you for accepting our invitation to deliver lecture. This is freight is very important topic for biologists who are working on protein interaction, biosensing, and other aspect also.
Yes.
So he did his PhD from IIT BHU, Banaras Hindu University, and MSc from Awadh, है ना? Awadh University. So with this brief introduction, his expertise is on this biosensing and freight mechanism. His background is chemistry, but he's applying that chemistry to the biological system. So you also attend this lecture attentively, Anna, with full focus, and you can interact as maximum as possible to learn, because sometimes topics are new, so it may be difficult for you to grasp 100%. But even if you are grasping 10-20%, that is enough. You will be 20% ahead than who are not attending. ये बहुत बड़ा भीड़ है, भीड़ में जो सीखता रहेगा वो आगे रहेगा, जो सीखेगा वो बढ़ेगा। फ्री का चाहेंगे सिर्फ सर्टिफिकेट लेना, या फ्री का मैंने बहुत टाइम में लोगों का डाटा भी है इसमें, है ना, आते हैं लास्ट में और फिर उसके बाद सर्टिफिकेट और ये सब। सो जस्ट ट्राई टू लर्न, सर्टिफिकेट इज बाय प्रोडक्ट, है ना। ट्राई टू लर्न एंड इट अप्लाई इट वेयर एवर पॉसिबल। सो आई वेलकम यू, अतुल जी।
Thank you, sir. Uh,
नाउ प्लेटफॉर्म इज योर्स।
okay, so I am sharing the slide.
Yes, same way you share entire screen and then your slide.
Is it visible, sir?
Just open your, yes, now visible, make it full slide. Yes, it is visible, very well visible.
Okay. So before discussing about the today's topic, thank you very much, sir, for inviting me and for your gestureful welcome. Uh, and I extend to my condolences in a for a tragic incident that happened today uh, to the families of deceased. So, myself, Dr. Atul Kumar Tiwari, currently serving as assistant professor in Uttaranchal University, Dehradun. So, during my PhD study, I worked on some surface functionalized metal nanoparticles and applied that nanoparticles for various applications like drug delivery. And fortunately, because my supervisor was actively involved in the electrochemical sensing of some
ये जो
metabolite.
यहाँ है उसको हाइड कर लीजिए, वहां जो एक लाइन आ रहा है ना,
हाँ,
उसको हाइड कर दीजिए। यस, यस,
okay.
प्लीज, प्लीज गो अहेड।
So, my interests are also diverted toward the biosensing of some sensing of some environmental contaminants as well as biomolecules. So, in a fortunate experiment, I applied the polyethylene stabilized metal nanoparticles via fluorescence energy transfer methodologies to detect some specific biomolecules. So, in this row, uh, I will discuss some fundamentals of thrust-based sensing, application of FRED technology, and further extend my talk with discussing some recent advancement in the field which has been done by me during my studies. So, in the first slide, first of all, I would like to explain the FRED technology. What is the FRED? So we know that in a phenomena when a chromophore interact with the light or radiation, the molecule get excited and return to fundamental state by emission of some energy. So light energy is absorbed by chromophores on a scale of 10 to power minus 15 second and emits energy that is called also fluorescence in a time scale of 10 to power minus 9 second. However, other possibilities are also associated that the excited molecules can transfer its energy after the excitation to the energy to a chromophore, a nearby chromophore without radiation or to another fluorophore, which in turn fluorescence in the same order of time that is 10 to power minus 9 second. So overall, this phenomena is called as fluorescence or Foster energy transfer. So in further, Fred phenomena is a purely physicochemical phenomena that is based on distance between the two fluorophore molecules. For Fred phenomena to happen, there is need that two adjacent molecules must be fluorophores that has the must have a capability to fluoresce under the radiation or light condition. So it entails quantum mechanical and non-radiative transfer of energy without photon emission. That is very important point. Without photon emission energy transfer is the fundamental property of FRET based technology. How? Via dipole-dipole coupling from an excited donor to an electron acceptor in its ground state. So here you can see that there is a Jablonski diagram where the electron from ground state absorb some energy and excited to S1 or S2 and revert and re-emit the energy via relaxing due to solvent and fluoresce in a time scale of 10 to power minus 9 second and get relaxed again. In the second figure, which is describing the basic principle of FRET, you can see in the figure 1 spectral overlap. So what is the prerequisite condition for FRET to be happen? So two fluorophores, one is called donor and second is called acceptor. When donor emission overlap with the acceptor excitation spectra, then FRET can be happen. But in case when donor emission and its receptor excitation spectra does not match, no FRET happen overall. In the yes, in this figure you can see that there is an overlap between the donor emission and receptor excitation. So FRET can be happen. Further, one prerequisite is the donor fluorophore and acceptor fluorophore must be in a certain distance. So theoretically, the distance must be between 3 to 10 nanometers. And practically, it is also possible to put the two fluorophores, that is called the donor and acceptor, in within a distance of 10 nanometers. So if the distance is more than 10 nanometer, no FRET happen. Again, the distance between the 3 to 10 nanometer, a FRET happens. And third prerequisite of FRET technology is the correct orientation, molecular orientation of the two fluorophores, that is donor and acceptor. So if both fluorophores are in opposite orientation or one is in correct orientation and second is in distorted orientation, no FRET will happen. So for FRET based energy transfer, the both donor and acceptor must be in a correct orientation. So essentially energy transferred through space. One fundamental thing in FRET is The energy direct transfer from donor to acceptor is through space rather than through the typical emission and subsequent reabsorption. So there is no medium required to transfer the energy. So this transfer of energy is only possible when donor and acceptor molecule are physically separated by 1 to 10 nanometer, as I discussed, followed by overlapping of their electronic energy levels and appropriate fluorophore molecule orientation. So FRET can be accurate measurement of polymolecular proximity at angstrom distance and highly efficient if donor and acceptor are positioned within a Foster radius. So what is the Foster radius? The Foster radius is the distance at which half of the excitation energy from donor is transferred to the acceptor. So Foster radius must be between 3 to 6 nanometer. Foster in 1948 has explained the FRET in terms of relative orientation of donor and acceptor transition, that is dipole-dipole interaction and distance between the two molecules. So I must here emphasize that the audience, if they understand what is the dipole-dipole interaction is a very best.
आप कहाँ पर हैं? किसी रोड साइड में हैं क्या आप? आपका क्वार्टर?
हाँ सर, थोड़ा गली है बगल में.
अच्छा अच्छा, बहुत हॉर्न बजाने वाले से बहुत सारे लोग.
Yes, I am sorry for that, sir, but
कोई बात नहीं,
I
प्लीज प्लीज
unfortunately
कंटिन्यू
I
करिए.
cannot do anything.
कोई बात नहीं, चलिए.
Uh, so uh, I suggest to go for dipole-dipole interaction. What is it and how dipole-dipole interaction established between the two molecules? Because every molecule is have a dipolar structure and charge separation on that three-dimensional structure of molecule. So dipole-dipole interaction is very crucial in FRET-based technology. So further, Foster described the efficiency of energy transfer by affected by these parameters as I discussed. First is the distance between the two fluorophore, orientation of the fluorophores, participating fluorophores, and dipole magnitude of dipole-dipole interaction. So energy transfer yield is given by equation as you can see here. The E equal to R6 R0 R6 plus R60. So either mathematical calculation you can very easily calculate the energy transfer efficiency whenever you do or we do the flood-based technology experiments. So what is R? R is the best Foster radius, which is the 50% energy transfer. So experimentally, the energy transfer efficiency can be obtained by monitoring the fluorescent intensities in the presence of donor fluorescence and acceptor fluorescence by the calculating the solving the formula 1 E equal to 1 minus FDA upon FD. So now I am coming to the application. I am not going very deep because a lot of and huge things are to describe the FRET based technology, and I suggest and recommend to all audience who are not very much aware with the FRET, go to the some standard books, and I am sure that FRET based technology is very uh, important to in today's biosensing field and protein and biomolecular studies. So here you can see the two circles that is uh, are emphasizing the membranes, a vesicles. Now, if we have to understand the membrane fusion and monitor the dynamics of membrane fusion by FRET-based technology, what we will do? So, we will do something like that, that one vesicle, we will tag the one vesicle with the donor acceptor fluorophores adjacent to each other, and one will keep the non-labeled. So when the two vesicles fuse with each other, but under non-fusing condition, the donor transfer energy to acceptor and a fret-based fluorescence emission happens. But after the fusion of the second vesicle, another vesicle, then the distance between the donor and acceptors get extended. So the fluorescence emission get disturbed. So overall, the fused vesicle have no fluorescence emission. Second, if we monitor, if we have to monitor another way, so put a fluorophore that is have capability to accept the energy, and another labeled with the donor. So after fusing of the vesicle, the donor and acceptor get close in the proximity and a fret happens. So based on these observation, very simple and crucial observation, we can apply the fret-based technology in the membrane fusion experiments, and some because uh, lipid nanoparticles and all the different type of lipid nanoparticles are today applied for the drug delivery purposes, and different type of modifications are also required to deliver the specific cargo at a very site specific manner. So these kind of experiments and validation are required. So FRET is the very best technology to monitor the membrane fusions. In the second example, how we can decipher the way how a protein or micromolecule uh, macromolecule change its conformation or shape. So, and further, we can also monitor the interaction of receptor with its ligand. Or so receptors generally occur at the membrane of unicellular or multicellular micro organisms. So when ligand bind with the receptor, so how we will monitor that? So FRET is the best technology to monitor the receptor and ligand interaction. How? When in a figure A, you can see that ligand is interacted with the receptor. Ligand has a donor fluorophore and acceptor has a receptor has a acceptor fluorophore. When the ligand interact with the receptor, a fluorescence emission happen. But when ligand get dissociated from the receptor, the intensity, fluorescence intensity get lost. In the second experiment, example, how we can monitor the conformational change in the in a intramolecular supramolecular structures. So we just put the donor and acceptor on a specific location in the micromolecular structure. So in a specific condition, the FRET happens. That means the energy transfer from donor to acceptor. But under conformational change dynamics, the donor and acceptor distance get extend, and FRET phenomena could not able to takes place. In the third example, substrate and product. So we can label the substrate with the donor receptor as it has, and plus enzyme. But when enzyme convert the substrate into product, the donor and acceptor distance get widen and fluorescence intensity get lost. So these are the fundamental examples of how we can apply the FRET technology in the protein-protein interaction, conformation, molecular membrane trafficking, membrane fusion, and monitoring the reaction kinetic enzyme with reaction kinetics. So all these things are can be monitored and tracked by FRET-based technology. So now coming to the most recent example, apart from protein interaction and protein detection, FRET is also extensively used for quantitative detection in PCR. In this technique, one of the PCR primer, because PCR start with the priming, so PCR primer is labeled with the donor fluorophore and the other primer is labeled with the acceptor fluorophore. Further, when the two primers bind to the target DNA sequence and in close proximity, the donor fluorophore transfer its energy to acceptor fluorophore, resulting in the fluorescence signal. Further, FRET can be used in real-time PCR to monitor the amplification of DNA. As PCR reaction progresses, the amount of as the DNA content increases, the fluorescence intensity increases inversely. So this allows for the quantification of DNA present in the reaction. It can also be called real-time quantitative PCR. During COVID-19, several researchers also performed the real-time PCR, real-time quantitative PCR used the FRET technology to quantify the amount of DNA. So for PCR, we can use the basic fluorophore dyes like fluorescein, rhodamine, and cyanine. However, based on the experiment, the choice of fluorophore can be different depending on the which kind of instrument we are using and what is the specific requirement for that particular experiment. So overall, we can summarize the application of FRET in various fields like assaying ions, small molecule, and micromolecules, binding studies. When the two molecules bind to each other or interact to each other, we can monitor their binding dynamics via FRET, like protein-protein interaction, protein-nucleic acid interaction, and nucleic acid interaction. Further, we can monitor the dynamics of protein and nucleic acid interaction and uh, other experiments. However, in detection of a small molecule, micromolecules, and detection of protein, nucleic acid, and conformational change are also very obvious to can be monitored by FRET technology. Nowadays, uh, FRET technology is being used in the live cell imaging, diagnostic approaches, drug assessment, and tissue-based analysis and cell-based assay. Now I am coming to the very crucial point, what I have done in my recent uh, research work, and also this is the topic of today's talk, role of molecular spacer and FRET-based biosensing. So, what are the spacers? First question is, what are spacer? Spacer molecules are a specialized molecules or macromolecules that put the donor and acceptor in a certain distance, that is 10 nanometer. So, spacers are very crucial or FRET-based technology because if donor and acceptor are not aligned in a specific distance, at a specific distance, no FRET will happen. So it play a very vital role in ensuring donor and acceptor physical position for efficient energy transfer. Therefore, investigating the impact of synthetic polymers polymer spacer in FRET has revealed several important factors. However, spacers can be polymeric or natural, and different various type of spacers are available, and the researchers are working with their own designed spacer molecule. But today I will talk because I have some experience in the polymer synthetic polymer spacer. So I will particularly talk on the polymeric spacers. So What are the fundamental characteristics of a molecular spacer and how we can manipulate the structural designs of the molecular spacers. First of all, it is important that size of a spacer is uh, must be very concise, that put the donor and acceptor in a close proximity, that is the Foster radius 3 to 10 nanometer. So optimum distance is required, for it is ineffective if the fluorophore are too far too apart, whereas excessive closeness can result in quenching or interference. Spacers must be flexible. Not must be flexible, some workers also worked on the rigid spacer, but the flexibility of spacer matters depending on the type of experiment. Polymers can be designed with either flexible or rigid backbones depending on the requirement of the biosensor. Third is biocompatibility of spacer. If we perform a in vivo sensing or imaging experiments via FRET technology, then spacer must be biocompatible. Like some common choices for biocompatible spacer molecules are polyethylene glycol and polylysines. Third is customization. Biosensor can be tailored to achieve high specificity by adjusting the length, flexibility, and chemical properties of polymer spacers. In protein-protein interaction studies, spacers can be customized to optimize the fluorophore distance based on the anticipated protein conformational change. Fourth important characteristic is precision and selectivity. Appropriate spacer length and composition can enhance the precision and selectivity of biosensor. Now, based on the some important features, we can classify the polymeric spacers in two major domains. One is rigid spacer and second is the flexible polymeric spacers. So I will talk first rigid spacers. So rigid spacers are vital component in field of chemistry, biochemistry and biophysics, particularly in areas such as FRET, molecular recognition, protein folding studies and drug development. They maintain a consistent distance between functional group, molecule, and atoms while restricting their relative motion. So, rigid spacers has a specific feature that it put the donor and acceptor in a restricted motion condition. It does not allow the motion of donor and acceptor. This rigidity of spacer is essential for precise spatial control, making these spacers crucial in scenarios that require specific orientation or distance between the interacting elements. So here is one very good example that has recently worked by a researcher in 2022, and they put a rigid spacer called bicyclo 222 octane cage spacer. You can go and read the article because references is provided here. So here you can see the spacer. Here is the donor molecule, here is the acceptor, and this is the spacer. So it is called the bicyclo 222 octane cage spacer. Further, several other rigid spacers are piperazine and biphenyl spacers. Workers are doing actively by customizing the spacer molecule. So in these experiments, you can see here that this is the piperazine spacer and here is the biphenyl spacer molecule, because here is the donor, here is the acceptor. Now I'm coming to the flexible spacers. Flexible spacers are integral to the development of FRET based on biosensor, including polyethylene glycol, polyproline, and polypeptides. Protein molecules or polypeptide macromolecules are also act as a spacer, flexible spacers. So depending on experiments, we can also apply the polypeptide as a stabilizing agent of the nanoparticles as well as the spacer molecule. So flexible spacers ensure the appropriate spacing and flexibility between the donor and receptor fluorophore, which is crucial for accurate monitoring of biological process. PG polyethylene glycol spacers are favored in biosensors because their flexibility, biocompatibility, and hydrophilic nature, which enhance the biosensor solubility for in vivo condition and stability, reduces non-specific interaction and improve performance. So here is the example of PEG flexible spacer that is worked by uh, some researchers. However, maybe I missed here the reference. By the way, so now I'm moving to the current scenario and threat-based biosensing. So the fluorophore, conventionally used fluorophore molecules has some shortcomings, like stability of fluorophore is a challenge. Further, its biocompatibility, we cannot use the synthetic fluorophore like fluorescein, rhodamine, and cyanine dye or in vivo application because they may be toxic. So, researchers currently are exploring the opportunity to introduce the nanoparticle mediated thread based biosensing. So, in that order, to achieve higher, greater efficiency and sensitivity, researchers are exploring alternate. So, various nanoparticles such as quantum dots, graphene quantum dots, and up conversion nanoparticles have demonstrated potential Fred donors, while gold nanoparticles are very potent acceptor nanoparticle. These materials are appealing because of their superior attributes, including enhanced efficiency, increased stability, and improved performance in biosensing application. And one most important thing is by tailoring the surface of quantum dot and graphene quantum dot or any other nanoparticle, we can reduce the toxicity, if any, of these nanoparticles for uh, bio in vivo applications. Further, among metal nanoparticles, gold are notable for their unique characteristics such as surface plasmon resonance effect, high conductivity, age of surface functionalization, and exceptional fluorescence quenching capabilities. Now I am coming to my work, which is recently published in a very good journal. So, role of different polymer stabilize. In this work, I have applied multiple polymers to assess their ability to be as molecular spacer in for stabilization of gold nanoparticle and their applicability in the glutathione detection via FRET-based turn-off, turn-on methods. So here you can see that gold nanoparticles are stabilized with the spacer that is, it may be 3-amino propyl trimethoxysilane, polyethylenimine, or polyindole molecule, or many maybe some other molecule depending on the experiment and lab setup and availability of the chemical. So anyone can customize the spacer molecule based on their requirement. So what happens? Actually, a fluorophore, that is a chemical, transfer its energy to the gold molecule, gold nanoparticle via non-radiative energy transfer. So here you can see that a spectral overlap is essential for ferret. So it must be between 1 to 10 nanometer. So why my work is important? First of all, glutathione is essential oxidant. It is very important to make a balance in the body to her a radical scavenger. It maintains cellular redox balance with fluctuations in level glutathione linked to cancer, cardiovascular disorder, and neurodegenerative disease. In recent years, researchers have developed fluorescent nanoprobes for biothiol detection using various nanoparticles such as silicon quantum dots, nitrogen doped quantum dots, and gold nanoparticle based system. Several other studies have also reported fluorescence techniques or biothiol analysis that utilize fluorescein and gold nanoparticle. So in this technique, which I applied, a fluorescence probe called fluorescein, which serve as fluorescent reporter, is quenched by metal nanoparticle, that is gold nanoparticle stabilized by polythylamine 3 amino 3 aptms and polyindol, act as a fluorescence quencher. So when we talk about the fluorescence quencher or fluorescence enhancer, some specific molecules make fret via enhancing the intensity of fluorescence, and some specific molecules or nanoparticle quench the intensity of uh, fluorescence of a specific fluorophore. So we can divide the technology by into two groups. One is by quenching and second is enhancing. So I will discuss here the quenching method. Further, I have worked also on the fluorescence enhancement. You can go and access the related paper on my Google Scholar or some other sources that I will provide in the last. So why my work is important? Because metal nanoparticles are stabilized with different chemical agents that actively participate in fluorescence quenching and enhancement. First of all, I have to discuss that why metal nanoparticles are stabilized with some specific molecules or polymers. So polymers provide a better stability to the nanoparticle because it prevents nanoparticle from agglomeration and continuous and slow leaching of the atoms uh, from the nanoparticles. So stabilization is very important. Various polymers are used for the stabilization of gold nanoparticle, silver nanoparticle, and quantum dots. You may explore the things on different research article, or you can try to explore the things on Google Scholar also. So therefore, in our study, we synthesized monometallic gold and silver, and bimetallic nanoparticles using different stabilizing agent as I discussed, indole 3 carboxylic acid, polythylenamine, and 3-APTMS, and investigated their ability for reporter fluorescein quenching and turn on sensing of glutathione. So in results, we found that polythylenamine, indole, and 3-APTMS are act as a effective stabilizer for synthesizing noble metal nanoparticles such as gold and silver. Polythylenamines are very branched structure, captures gold cation with amine group, following by reducing and nucleating of gold cation. Further, polythylenamine yields a well-dispersed gold nanoparticles due to strong repulsion. Because polythylenamine are cationic in nature, so it provides overall cationic charge on the stabilized nanoparticle that promote the strong repulsion between the two nanoparticles and prevent the agglomeration of nanoparticles via forming a thick Pi layer, polythylenamine layer on nanoparticle surrounding. In aprotic solvents like acetone, indole monomer form a acid-base adduct and metal cations triggering 1,3 polymerization and nanoparticle formation. So the first time we are also introduced the indole as a reducing as well as stabilizing agent of gold nanoparticle. Further, indole act as a reducing promotes slow redox reaction by reducing the gold cation to metallic gold and oxidizing agent to polyindole. These simultaneous reactions allow gold nuclei to grow with a polymer influenced by the medium polar component because of acetone. Further, one interesting outcome which we have got that before my entry in my laboratory used my hot air oven to produce the nanoparticle, which take at least 6 to 8 hours. But one day, fortunately, I tried the kitchen microwave oven to produce a large amount of highly reduced nanoparticle in a very short time. I can say it is after exposure of microwave, the gold metal cation converted into nanoparticle within 30 seconds with a very good efficiency of reduction. So this point is also important to note down that how microwave processing influence the reduction process and fasten the reaction kinetics in the conversion of nanoparticle. Further, to examine the effect of capping and stabilizing agent on fluorescein reporter quenching and release infrared-based glutathione sensing, silver, gold, and silver gold bimetallic nanoparticles were created. So here is the mechanism how I created different type of stabilized and functionalized monometallic as well as bimetallic nanoparticles. I am not going very deep. You can read the article. Uh, it is available or internet. Yes. So I characterize the metal nanoparticles which synthesized. So here you can see that five type of nanoparticles having different stabilizing agent and absorption maxima, further followed by the differences in zeta potential. Here you can see the gold silver bimetallic nanoparticle both peaks because silver have a characteristic peak around 405 and gold has a very specific SPR characteristic at 520 nanometer. It is theoretically, however, experimentally it could be depending on the type of functionalizing or stabilizing molecule, type of polymer which we are applying for the stabilization. So depending on the personal choose and experimental setup, the standard SPR characteristic of gold and silver can be changed. So in my case, the bimetallic nanoparticle have shown the two peaks. One was at 425, that is characteristic of silver, and another is 440 nanometer, 540 nanometers, that is the characteristic of gold. And you can see that there is a core cell structure having gold nanoparticle in the core and silver nanoparticle on a cell. Further, I have stabilized silver nanoparticle with the PI via microwave processing, which showed a SPR characteristic at zero point 0.2 nanometer absorbance. Further, similarly stabilized with 3 APTMS silver nanoparticle shown at SPR at 0.0407 nanometer. Polyindole stabilized gold nanoparticles presented demonstrated the absorbance at 504 nanometer. Polyethylene stabilized gold nanoparticles demonstrated absorption at 5 to 12 nanometer. Similarly, based on the stabilizing agent, that is polymer, the zeta potential of the synthesized nanoparticles were accordingly. PI stabilized bimetallic nanoparticle had a zeta potential of 19 plus 19 millivolt. PI stabilized silver nanoparticle had a 20 plus 26 millivolt. 3 APTMS stabilized silver nanoparticles was around 15 millivolt, while polyindole stabilized gold nanoparticles was minus 15 millivolt. Interestingly, the gold nanoparticle stabilized with polyethylenamine have demonstrated a very high zeta potential in plus region. It was around plus 32 millivolt. That signifies the stability of nanoparticle due to the strong nanoparticle repulsion and least chance of agglomeration between in nanoparticles. Here is the TEM characterization. You can see the core cell structure of bimetallic nanoparticle. It is very clearly visible that core and cell structure. Further, uh, these are the different nanoparticles are stabilized. Here is the PI stabilized. The figure E is demonstrating the PI stabilized gold nanoparticles. You can see that nanoparticles are very well dispersed in comparison to uh, B, that is 3 APTMS stabilized silver nanoparticle, and C, polyindole stabilized gold nanoparticles. PI stabilized gold nanoparticles were very well dispersed and least chance for agglomeration. So here the mean size PI AZNP silver PI stabilized gold silver nanoparticle was high in size, that was around 22 nanometer in actual size, not hydrodynamic radii. Hydrodynamic radii is different thing and actual size is different parameter. However, polyethylenamine stabilized gold nanoparticle was around below 10 nanometer or around 6 nanometer in size. Now I am coming to some findings. Here you can see the dashed line is fluorescence emission of PI stabilized gold nanoparticle. Solid black line is the UV visible spectrum of PI stabilized gold nanoparticles, that is around 512 nanometer, and the red line is indicating the emission of fluorescein molecule. So here you can see the spectral alignment was exactly demonstrating and showing the possibility for dipole-dipole interaction between the fluorescein molecule and gold nanoparticles to conduct a FRET-based sensing methodology. So based on, because gold nanoparticle is yes, fluorescein molecule is emitting at height 20 nanometer, while gold nanoparticle is absorbing on height 12 nanometer of wavelength. So there was a very best platform to study some biomolecules or sensing of some biomolecules like glutathione. However, the dynamics of glutathione interaction with the fluorescein metal nanoparticle system is somewhat different, which I will explain in the next slides. So I tried to explore that what was the fluorescent, uh, fluorescence quenching efficiency of different synthesized nanoparticles. So here you can see the PI stabilized bimetallic nanoparticle. This y-axis is the fluorescent quenching, and axis is the different type of nanoparticles. So PI stabilized bimetallic nanoparticles quench around 55% of fluorescein emission, while polyindole quench around 97%. PI stabilized gold nanoparticle quenched around 94%, and PI stabilized silver nanoparticle quench around 80% of fluorescein emission, while 3-aminopropyl trimethoxysilane stabilized silver nanoparticle was quenched negligible. There was no dipole-dipole interaction, so quenching was not possible in this case. Here is the uh, chromaticity coordinates of the different nanoparticles. It was in blue region. Uh, so further, I tried to find out whether,
और कितने स्लाइड बचे हैं?
sir, now I'm moving very fast, sir.
हाँ, फास्टली, क्योंकि ये केमिस्ट्री, डीप केमिस्ट्री तो बायोलॉजी वाले को समझ में भी नहीं आएगा ज्यादा।
नहीं नहीं सर, बायोलॉजी पे ही आ रहा हूँ मैं अब।
अच्छा अच्छा, हाँ, क्योंकि एक घंटे के लिए इनको
Because
बोलते हैं तो।
very important, and I will explain the things with some graphical
हाँ हाँ
data.
हाँ हाँ हाँ, जल्दी कर दीजिए, है ना।
So, what was the plausible mechanism uh, for turn off and turn off detection? So, PI stabilized bimetallic nanoparticle quenched the fluorescein via dipole-dipole interaction with 53%. However, after the addition of glutathione in the fluorescein and metal nanoparticle system, no fluorescence recovery uh, happened due to the presence of silver on the cell. So before this, I would like to provide an information that gold glutathione, because glutathione carry a thiol group that has a very strong capability to interact with the gold atom as compared to silver. So due to the presence of silver on the cell, the glutathione does not interact with the nanoparticle, so there was no fluorescence recovery. Further, similarly with polyindole stabilized gold nanoparticles, quenching was very good, but due to but after addition of glutathione, no fluorescence recovery happened because the polyindole molecules was very strongly adhered to the nanoparticle surface, and glutathione does not get any access to interact with the gold nanoparticle surface. So there was no chance for the gold and sulfur interaction. Similarly, PI, as I discussed, quenched, but no recovery after the addition of glutathione, because most of the glutathione, due to the least affinity of thiol group for AGNP, and 3-APTMS stabilized silver nanoparticles are not very well quenched. But you can see, interestingly, you can see the PI stabilized nanoparticles quench very well the emission of fluorescein, but after addition of glutathione in the fluorescein metal gold system, a linear increment in the fluorescence takes place. That you can see here. Further, Further, here is that very dispersed nanoparticle, but after addition of glutathione, the nanoparticle get agglomerated. So, what is the mechanism behind why this kind of phenomena is happening? So, you can also look at the 3D fluorescence spectroscopy. Here is the fluorescein emission after addition of gold nanoparticle, PI stabilized gold nanoparticle, and further addition of glutathione. It increases the quench fluorescence intensity in a linear manner. Further, after adding another quantity of glutathione, the fluorescence recover of the system. But what is the plausible mechanism behind it? Because polyethylene stabilized gold nanoparticle was in size around 5 nanometer and having a zeta potential plus 32 millivolt. And similarly, fluorescein had a excitation emission at 490 to 520 nanometers and zeta potential of minus 36 millivolt. So minus 36 millivolt and plus 32 millivolt promoted a very strong dipole-dipole interaction or electrostatic interaction between the fluorescein molecule and gold nanoparticle, which promoted and paved the way for the fluorescein quenching, because the gold nanoparticle is a very good acceptor of the fluorescence emission. But after the addition of glutathione molecule, a ligand exchange takes place at the pi stabilized gold nanoparticle surface by loosely bound polyethylene molecule, which lead to the gold sulfur bond formation. AUNPs get agglomerated, followed by fluorescein molecule free to fluoresce again. Here you can see the gold nanoparticle get agglomerated. Here you can see the after addition of glutathione, the nanoparticle get agglomerated. So what was the significant finding? Polyethylenimine and polyindole molecules were acted as an excellent spacer, which keeps the donor fluorescein and acceptor gold nanoparticle core in a close proximity. But still, there is a chance to understand and further study what are the molecular orientation of fluorescein and gold nanoparticle, and what are the condition at which the dipole is establishing between the fluorescein and gold nanoparticle, except the zeta potential and size of the nanoparticles. Why pi is a excellent spacer molecule? No inherent absorbance, because pi absorbs very less uh, UV or visible light, and help in detection and complication. Application of pi is, and these two properties it makes and useful for application where you want to study the interaction of pi with other molecule by observing changes in absorbance when they complex together. So I published these results in MRS communication, while the impact factor is very less, but journal is very reputed, and peoples are looking very well towards our work. So thank you very much. Here is my research team. I thanks to Professor Pandey and Professor Rojan Narayan from Joint Department of Biomedical Engineering, United States, for their consistent support and uh,
थैंक यू,
support
थैंक यू
and providing the
अतुल तिवारी जी, बहुत बढ़िया।